Hello friends, welcome to another video of our rice disease series. So we have already discussed many important diseases of rice like starting from the rice blast, rice brown spot, bacterial leaf blight of rice. Now we will discuss a major viral disease of rice, the Rice Tungro disease. So in the various competitive exam, definitely you will get one question from the Rice Tungro. This is that important. Okay, because of its complex nature, this is mostly asked. So just like the previous videos, here also it contains three sections. So in the section 1, MCQs question will be there. In the section 2, SSR reasoning type questions, and in the section 3, quick revision points will be there. So I don't think you need to go through any books or anything to study this Tungro disease. If you go through the quick revision points, everything is mentioned there, so you will get a thorough idea about the Rice Tungro disease from this video. Okay, so without further delay, let's start the video. So the question number 1, Rice Tungro disease is caused by. So the options are option A, RTBV alone, option B, RTSV alone, option C, both RTBV and RTSV, and option D, phytoplasma. So this is caused by RTBV and RTSV alone. Sorry, RTBV and RT, both RTBV and RTSV. So we will describe it in the other later questions. Okay. So the question number 2, RTBV stands for. Somewhere I have already seen this question in some competitive exams. Okay, what RTBV stand for and RTSV stand for. So the options are option A, Rice Tungro Bacilliform Virus, B, Rice Transmitted Bacilliform Virus, option C, Rice Tiller Bacilliform Virus, D, Rice Tropical Bacilliform Virus. So here the answer is option A, Rice Tungro Bacilliform Virus. Similarly, the question number 3, RTSV stands for Rice Tungro Spherical Virus. So here two variants of Rice Tungro disease are there, Rice Tungro Bacilliform and spherical. So these two virus, they actually, when they are co-infecting a plant, that is leading to the Rice Tungro disease. So Rice Tungro disease is caused by co-infection of Rice Tungro Bacilliform and Rice Tungro Spherical Virus. Why these are named differently? Because they are different in shape and size. This is bullet shape and this is spherical. Not only shape, in terms of genome also they are different. We will discuss later. Okay. So this is about the virus. Then about the genome. So the genome of RTBV or Rice Tungro Bacilliform virus is option A, single stranded RNA, option B, double stranded RNA, option C, circular double stranded RNA, or option D, linear double stranded RNA. So here the answer is the genome of Rice Tungro Bacilliform. If it is Bacilliform, then it contains the double stranded circular DNA. Okay, here the DNA, this is a DNA virus, double stranded circular DNA. Then what about the genome of RTSV or Rice Tungro Spherical Virus? Option A, polyadenylated single stranded RNA, or option B, the double stranded DNA, option C, double stranded RNA, or option D, viroid RNA. So here the answer is for the spherical virus, the genome is polyadenylated single stranded RNA. So remember this, for Bacilliform it is double stranded DNA virus. The genome is in the form of double stranded DNA. For the Rice Tungro Spherical Virus, for the spherical form it is single stranded RNA. Okay, these are very important thing, remember this. Next, The major Tungro symptom are caused by. So this virus disease, actually this Tungro disease is caused by the infection by two viruses. But the major symptom caused by which virus? So the options are option A RTSV, option B RTBV, option C both equally, and option D vector toxin. So here the answer is the major symptoms are produced by option B RTBV. The Bacilliform virus, Rice Tungro Bacilliform virus, is responsible for the production of major symptoms of Rice Tungro disease. What symptoms it produce? So generally yellow to orange discoloration will be formed. If this is the leaf, so discoloration generally start from the tip or the margin of the leaf. So this is the leaf, means discoloration will be observed, yellow, that is of yellow to orange color discoloration. That is also specific in which cultivar this orange color discoloration is mostly observed and where the yellow color discoloration is observed. We will discuss everything in detail. Okay. Next, question number 7. Efficient vector transmission required. Option A RTVV alone, option B RTSV alone, option C RTBV plus RTSV, and option D fungal association. So here the answer is for efficient vector transmission, means which virus is responsible for the transmission, that is the RTSV alone. So two things you got here, that the symptom portion, this part was mainly regulate or mainly imparted by the Rice Tungro Bacilliform virus, whereas the transmission of virus are performed by the Rice Tungro Spherical Virus. So if the spherical virus, Rice Tungro Spherical Virus is not there, then this Rice Tungro Bacilliform virus can't transmit from one plant to another plant. So for transmitting through a vector, this helper virus, Rice Tungro Spherical Virus, is necessary. It is responsible for the transmission. Okay, but if this RTSV is alone present in the host, this RTBV is not present, means the symptom will be very mild or no symptom will be there. For the symptom expression, this RTBV is important. The bacilliform type is important. Remember these things. Okay. Next, the question number 8. Characteristic symptoms is for the Rice Tungro disease, obviously. Option A yellow-orange discoloration, option B water soaked lesion, option C small balls, or option D white hair. I have already told you that the option A is correct. Yellow to orange discoloration starting from the tip or the margin of the plant leaf. Okay. Then question number 9. Yellowing in Tungro starts from, this also I have already told, start from the leaf tip. Okay. Next, question number 10. A conventional diagnostic test for Rice Tungro disease. Options are option A gram stain, option B iodine test, option C ELISA only, option D KOH. So here they are asking the conventional diagnostic test, not the advanced. If it was called it was asked advanced test, you can mention the ELISA, but here they are asking the conventional diagnostic test for Tungro, that is the iodine test. This is very important. So in generally, so what happened, in the morning time the leaf will be clipped and it will put on the iodine solution. So ultimately for the positive, this is a starch test. So if it is Tungro positive, then it will discolor, means some bluish color will form. Okay. So same question it is given here. That question number 11. Positive iodine test shows, means what color of discoloration it will, or color it will appear. I have already told, dark blue streaks. So when we dip the leaf infected with Rice Tungro virus in a iodine solution, it will produce dark blue streaks. Next, question number 12. Rice Tungro Bacilliform Virus particle are option A spherical, option B bacilliform, option C filamentous, and option D geminate. Obviously, bacilliform virus means it is obviously bullet shape or bacilliform type. Then question number 13. Rice Tungro Bacilliform particle dimension are approximately. So option A 130 into 30 nm, option B 30 into 30 nm, option C 300 into 30 nm, option D 50 into 20 nm. So here the answer is 130 into 20 nm. So you can use a trick here that the bacilliform in the I have also started a virology series. There we have uploaded a video on the architecture of virus. You can check that. So 3 type of architectures are there: spherical, helical, icosahedral, and this complex symmetry or the this bullet shaped virus. So in the bullet shaped virus or bacilliform symmetry type virus, what happened? The shape of virus, the virus are generally its length. It is of bullet shape, and its length is 3 times of the diameter of the virus. Okay, if it is 1, then this will be 3. So you can guess from this that if this is a bacilliform, definitely the diameter will be 3 times of the length will be 3 times of the diameter, which is possible for this option only. So you can guess like this. Okay, this is a trick. You can use this wherever it is if it's possible. Next, the question number 14. The RTSV or Rice Tungro Spherical Particle are. Similar easy question. It is obviously Rice Tungro Spherical Virus means obviously it is spherical in shape. Next, question number 15. Diameter of Rice Tungro Spherical Virus is about. This is a typical question. Okay, so you have to remember the answer. So here the options are option A 10 nm, option B 20 nm, option C 30 nm, and option D 60 nm. Here the answer is 30 nm. So bacilliform particle shape, obviously it is a bacilliform, is a bullet shape. So length and diameter both will be there. So here the dimension is about 130 to 30 nm. But here for the Rice Tungro Spherical Virus, it is 30 nm only. Next, question number 16. The most important vector is means for the Rice Tungro virus. Here the options are option A Niraparvata lucens, option B Nephotetex virescens, option C Bemisia tabaci, option D Myzus posicki. So here the answer is green leaf hopper or Nephotetex virescens, green leaf hopper. Okay. Next, question number 17. Tungro is transmitted in A. Option A persistent manner, option B circulative manner, option C non-persistent manner, and option D seed-borne manner. So this is generally transmitted in non-persistent manner, means it will not transmit from one generation to another generation, or neither it multiply inside its vector. So in the virology series we will discuss in detail about the vector transmission of plant viruses. So check out that playlist. That is very important. Within 10 days we will finish the whole unit 6 as per the syllabus given for the ARS net or the SMS STO by the ASRB. We will finish the virology syllabus. So check out that videos. Next, question number 18. Severe epidemics occur where? So here the conditions are given which are responsible for the severe epidemic development for Rice Tungro. Option A, rice is continuously cultivated. Option B, frost is common. Option C, vector absent. Option D, no wheat occur. So here the option A is correct. If the rice cultivation is continuously there, then only the severe epidemic will occur. Why? Because virus is a obligate intracellular parasite. It needs its host, and its host specificity is very high. So if rice cultivation is not continuous, it was not the host is not available throughout the year, it will die, the virus will die, and no such epidemic will be there. So for the epidemic development, continuous cultivation of rice is the must case. Next, question number 19. Important inoculum source is. So here basically they are asking the primary source of inoculum. So the options are infected stubble, option B collateral host, option C volunteer rice, and option D all of this. So here the option D is correct. All of this, infected stubble, collateral host means the weed host, and volunteer rice plants. These all are acting as the primary source of inoculum for the Rice Tungro virus. Next, question number 20. Which weed act as reservoir of host? Reservoir of the virus actually. Sorry. What happened? Okay. So reservoir for the, this is the virus actually. Okay. Reservoir for the virus. So here the options are Echinoclea colonum, option B Brassica campestris, option C Cicer aretinum, option D Gossypium. As we all know that this is a rice pathogen, and the collateral host will be rice host only, the Poaceae family host. So whichever are belong to the Poaceae family, that can act as, or the which are associated with rice field, that will only act as the collateral or weed host for this virus. So this is the Echinoclea colonum, which act as weed host for the Rice Tungro virus. Next, question number 21. Yellow discoloration due to Tungro infection mostly observed in option A Indica cultivars, option B Japonica cultivars, option C both, option D none. This is very important. So the yellow discoloration, generally for Rice Tungro, either yellow to orange, or in some cultivars specific yellow discoloration are observed, and in some cultivars specific orange discoloration are observed. So yellow discoloration generally observed by Tungro infection mostly in the Japonica cultivar, whereas the orange discoloration observed in case of Indica cultivar. You might have read the cultivars of rice in, these are the eco something physio, three types of cultivars are there for the rice. The Indica cultivars, Japonica and Javanica. Okay. So this Indica cultivars generally shows the orange discoloration, and Japonica cultivar shows the yellow discoloration. These things you should only remember. Okay. Next, question number 22. Which statement is correct? Option A, RTBV alone transmit efficiently. Option B, RTSV assist transmission. Option C, RTBV is RNA virus. Option D is RTSV is RNA virus. So here option B is only correct. That is the RTSV is assist the transmission. Everything I have already discussed. Okay, I don't think I will need to explain any of the option, because RTBV alone can't transmit because it needs the RTSV, and RTBV is a DNA virus, double stranded DNA. Okay, and RTSV is not DNA, it's single stranded RNA virus. Next, question number 23. Recommended nursery treatment is option A carbofuran granules, option B mancozeb, option C sulfur dust, or option D borax mixture. Here the answer is carbofuran granule. For the nursery treatment of for the management of Rice Tungro disease, carbofuran granules is used for the management of vector. We can't manage the fungicide, or we can't manage the virus with fungicide or the insecticide or any chemicals. We have to manage the vectors only. Okay. Then the question number 24. Monocrotophos is sprayed to control. Option are fungi, bacteria, leafhopper vector, nematode. As you all know that monocrotophos is a insecticide, means it is used to control the leafhopper vector, the GLH. Okay. Next, question number 25. Best long-term strategy is option A, disease tolerant cultivars, B, fungicides, C, antibiotics, D, soil solarization. So for the long-term strategy, if you are going, then it is the use of disease tolerant cultivars. Next, question number 26. RTSV means Rice Tungro Spherical Virus contain. This question is actually repeated. Here also answer is the option A, polyadenylated single stranded RNA. Next, question number 27. Which is not a Tungro vector? Option A, Nepotetis nigropictus. Option B, Nepotetis parvus. Option C, Roselia dorsalis. And option D, Scirpophaga incertulus. So all these three are efficient vector for the Rice Tungro virus. Only Scirpophaga incertulus. This is a insect that actually cause Scirpophaga. This is scale insect, I think. This is an insect actually, means it is harmful for the plant. It doesn't transmit the disease. These three are efficient vectors. Next, I forgot actually its common name. This is a very important insect, harmful insect for the rice. Oh, okay, you can just check it. Next, question number 28. Disease severity depends mainly on option A, host only, option B, vector only, option C, host virus vector interaction, option C, pH, soil pH. Here the answer is option C, host viral vector interaction. For the most of the viral diseases, the combination host virus vector interaction is most important for the disease severity. Next, question number 29. Integrated disease management emphasizes option A, vector control and inoculum reduction, option C, fungicide, option D, vectoricides alone, option D, deploying. Obviously it is the IDM management for the Tungro is for the vector control and inoculum reduction only. Then 30. A Tungro tolerant cultivar listed in notes is, so option are MTU 1002, option B TN1, option C IR8, option D CO43. Answer is MTU 1002. This is a Tungro tolerant cultivar. Then section number 2, Assertion reasoning type, then we will discuss. So quickly we will discuss. So first, the assertion, RTBV cause most Tungro symptom. Yeah, this is correct. Then reason, RTSV mainly facilitate transmission of rice, transmission by leafhopper. Both A and R are true, and R is correct explanation of A. Then second, the assertion, Tungro epidemic are severe in continuously cultivated rice ecosystem. Yeah, obviously it is true. And reason is host and vector persist throughout the year. Yeah, so both these are correct and it is true explanation of A. Next, third, assertion, iodine test can be used for diagnosis, obviously for the Rice Tungro. And reason, infected leaves show dark blue streaks after treatment. Yeah, both are correct and R is correct explanation of A. Then assertion 4, Rice Tungro Bacilliform alone is efficiently transmitted by vector. No, it is not transmitted alone efficiently. The Rice Tungro Spherical is not required for transmission. Both are false. Okay. This R and A both are false. Then question number 5, assertion, infected stubble contributes to disease carry over. Yeah, this is true. Then reason is the act as inocular source between season. Yeah, this is true. So both A and R are correct and are correctly explaining the A. Next, question number 6, assertion, leafhopper are major Tungro vector. Yeah, this is a major vector. True. The reason is transmission occur in a non-persistent manner. Yeah, this is transmitted in a non-persistent manner. So both A and R are true. Then 7, assertion, carbofuran help in reduction of disease incidence. Yeah, this is true. Generally in the nursery treatment, this is used. And for the field foliar spray, you can use imidacloprid, phosphamidan, edifenforsum, and insecticides are there. You can use that. Reason, it suppress vector population. Yeah, carbofuran is mostly used for the suppression of vector population. So option A is correct. Then question number 8, Assertion Tungro increase tillering. No, it reduced tillering. The disease caused stunting. Okay, reduced tillering. So it is false. Then reason, the disease caused stunting and reduced tillering. Reason is correct, but not the assertion. Next, question number 9, assertion, RTSV contains circular double-stranded RNA. Yeah, this is false. RTSV contain single-stranded RNA. Reason is RTSV possesses polyhedronism. The reason is correct, but this assertion is false. Next, question number 10. Assertion, disease-tolerant cultivar are useful in management. True for the Rice Tungro virus. The reason is they reduce yield loss caused by Tungro. Yeah, both are correct. Generally, the tolerant cultivar used because they reduce the yield. We reduce the yield loss. Even if the infection is there, the disease is there, still the tolerant cultivar actually manage itself. Either they enhance the yield or something. The physiological changes they use, mechanism they use to reduce the yield loss. They compromise the losses caused by the disease actually. So this is about the second section. Then quick revision points. So RTBV plus RTSV. So the co-infection caused the Rice Tungro disease. Then RTBV is for symptom and RTSV for the transmission. And then the RTBV genome is circular double-stranded genome, whereas RTSV genome is single-stranded RNA. RTBV particle size is 130 to 30 nm. It is of bacilliform shape. And RTSV is spherical shape with 30 nm diameter. Then the characteristic symptom for this is yellow to orange discoloration, which is in which orange discoloration mostly observed in yellow discoloration mostly observed in Japonica cultivar, whereas orange discoloration mostly observed in Indica cultivar. Next, the yellowing start from the leaf tip portion. This is characteristic thing. Okay, you have to remember this. Then the conventional diagnostic test used for the detection of Rice Tungro disease is iodine test, which gives dark blue streaks when the plant is Rice Tungro positive. Then reduced tillering and stunting occur due to the infection, and the primary source of inoculum is infected stubbles. Apart from this, the volunteer rice and collateral weed host. These three are primary source of inoculum. Then major vector is Nepototis virescens or GLH, green leaf hopper. Transmission is non-persistent manner. And management strategy mostly uses vector control through the insecticide or some other cultural management practices. And for the virus management, we can use the tolerant cultivar because no such resistant cultivar are reported for the virus. So tolerant cultivar can be used. So this is all about the Rice Tungro virus or Rice Tungro disease. I hope it will be helpful for you people. So thank you for watching.